Hey everybody, Mr. Kilmnick here. We haven't seen each other in a while, but there were a couple of things I wanted to go over. I know I just kind of threw out PowerPoint notes at you guys, but in this PowerPoint on serology, which is the study of blood, there were a couple of things I just wanted to clear up. I looked over the homework assignment, and I noticed a couple people got some things a little confused. So I'm just going to try and clear up those few things. Then this should pretty much be the only video that we will see for this unit. So first off, blood, what is it? It's 55% liquid, 45% solid. If you take a close look at this picture here, you could kind of see plasma, which is the liquid component. This was a blood sample. It was put in a centrifuge, spun around, and the solid part settled to the bottom, the liquid part on the top. And you could see 55% of blood is plasma. That is the liquid component. If we actually slide over now and take a look at the next slide on the PowerPoint, these are the cells down here. That's the other 45% of our blood. Those are solids. We find three types of cells: red blood cells, also called erythrocytes. They carry oxygen. That is their function. White blood cells, also called leukocytes. Their function is to fight infection. And then we have platelets in our blood, also called thrombocytes. That function is for clotting, so we don't bleed out. When you get a scab or something and you're bleeding, it's those platelets that are helping you out. A couple of other things that I wanted to look at. We saw in the PowerPoint antigens and antibodies. So what are they? Basically, an antigen is something that's foreign in your body, and basically that antigen, we call it a protein on the surface of the red blood cell. And really what we want when we are fighting off infections is for antibodies to actually fight that off and bind with the antigen. So that's really the function of those things. Now, I know another question you got was something called agglutination. Agglutination would be when your blood actually clumps up. So red blood cells get clumped up, and that happens when different blood types are actually mixed together. So if somebody's going to get a blood transfusion, they always need to know what blood type the person is, because you never want to mix those blood types. Once you do that, the blood can agglutinate and clot, and that can be very destructive for somebody. It could cause stroke or something very bad. So, taking a look now at the next slide, you could see there are different blood types: A, B, AB, and O. Those are our four blood types. And this just kind of shows what happens if you add the anti-A antigen to blood type A. What you see here, we got a problem, because once this happens, you will get agglutination. I'm sorry, not here. You'll get agglutination right here. So that's what it's showing on these pictures. Again, you do not want to mix blood types. All right, and this chart kind of shows all that. The most important thing I would say on this chart is, first off, if you have blood type A, you have A antigens, and the antibodies will be anti-B, because remember, the antibody is trying to fight off any foreign cells. So that you want anti-B, so the B cannot mix with the A. All right, and you could just study this chart pretty much and figure all that out. Now, frequency. The most common blood type is O, and the least common type is AB, right off the chart. Okay, so we got that. Now, let's talk about how we get our blood types. To figure out blood type, we use what's called a Punnett square. And a couple things you want to know. We get these alleles or genes from our parents. A and B are dominant alleles, and O is recessive. So in order to have type A blood, what happens is this. I'm going to slide down here for a second. Let's do this Punnett square. This is where I saw people getting a little confused. A man and a woman both have type AB blood. So, to make this the man, make that the woman, doesn't matter which way you do it, could have done it the other way. Determine the probabilities of blood types of the children they can produce. So, you see the 4 squares here? All you're doing in the squares is you're going to combine this allele with that allele. You're just matching them up. Here would be A and B from, I think, did we say dad or mom up here? I don't know. We'll make that dad. We'll make this mom. Okay, so there's dad, there's mom. Dad for here is giving A, and mom is contributing the B allele. And over here you could see BB. So here's the question. What is the probability of each blood type? So remember, I told you type A blood would be AA or A mixed with O. And the reason is A is dominant over O. So if A combines with O, this will win out. You'll have A-type blood. Type B blood, you need either 2 B alleles or, BO. And again, B is dominant, O is recessive. So if these two combined, your blood would show the dominant allele. For AB, A and B combined, and the only way that you can have type O blood is to have O and O match up. So let's take a look down here and let's see what percent. Well, there are 4 boxes. So each box accounts for 25%. I'll start with AA, and AA gives us type A blood, right? AB gives us type AB, and BB gives us type B. So let's see. This is the only one out of all 4 of these that would be type A blood, so 25%. AB, there are 2 boxes. 25 plus 25, that's 50. And 1 box left for BB, which is type B. And you could see, you could figure it out by adding up. They should always add up to 100%. All right, so hopefully that helps. And if you had any problems with the worksheet today, feel free to email me, and hopefully this clears things up. So everybody stay healthy, stay in, and if we need another video, I can always put one out. All right, talk to you soon.